Hello, good evening, friends. Welcome to today's topic. How can a gene therapy can cure hemophilia A? So, friends, here we will discuss in our pattern. Before that, let me show some disclaimer here. Friends, in this channel, I am going to discuss. I'm going to discuss topics from the Hindu, Telangana Today, the Financial Express, and Pioneer, and a small intro about myself. So, friends, I am Satya ji. You can find some books written by me on Amazon. Myself, I'm an ex-UPSC aspirant, and small disclaimer for you. If you find value from this channel, you can consider following me. And start away, we will jump into the paper. Yes. So, friends, this article came in the Hindu 15th December 2024. So, let me glance the paper. So, what we should capture for UPSC relevant. So, you should need to understand why this article is in news. So, here they mentioning CMC Vellore, they have some breakthrough. So regarding to hemophilia A, so it is a hereditary condition. So right, that you should check, and what is the usual treatment, and what is the cost for the treatment, and what is the technique they are imposing in this technique, right? So they are mentioning how the gene therapy. So they are mentioning how US F1D administration approved gene therapy for hemophilia, that is known as a Roctavian. And after that, what is the difference from the that Roctavian to hemophilia treatment in the Vellore? So there is a slight difference. We will discuss in detail there. Then comes to it is affordable, it is not affordable. So that is the small glimpse about how they are discussing in the article, but we should go in detail for our UPSC preparation. So it is not correct to read and leave itself. Got it? So we will discuss as it is in our pattern. So let me zoom some more. So this is our pattern, right? So yes, move to the concepts. So I hope it is visible for everybody. If not, let me know. Yes. Yes, friend. So let me deep dive. So friends, we all know we are learning the concepts, the fixed format. So that is this is the analysis pattern. First heading is why in use. So CMC Vellore, Christian Medical College of Vellore has some breakthrough with regard to hemophilia trials. So that is the concept. So what is hemophilia? Anybody, you can think about it for a moment. Yes, friends. Anybody can think about it for a moment. So friends, hemophilia, it is a rare and hereditary disease. What happens to human body if it comes, if the disease comes? Our blood will not clot immediately, will not clot. Blood flows flows outside very quickly. So loss of blood leads to other complications. So that's why it is very important to clot immediately when the blood comes out. So what is the general treatment? So friends, if you see, that is the replacement therapy. Replacement therapy they are mentioning, right? In the replacement therapy, they inject clotting factor. They inject clotting factor. Clotting factor 8. So that is the general therapy. So nowadays we have gene therapies, right? This is the main concept. And we have gene therapy. So we will see what is this gene therapy, because it is the main topic for the UPSC examination, and it is more in the current affairs in nowadays, right? So before that we will see some fundamentals, friends. So in the concepts we will see some fundamentals. So in the fundamentals we will discuss about some DNA. DNA is a deoxyribonucleic acid. It is a double helical structure, friends. Everybody, I think everybody knows, right? So it will be like double helical structure. So that you should keep in mind. And DNA is a information storing. It stores information storage. So that you should know. And another concept is, because we are going to apply the techniques on the either DNA or chromosomes. That's why you should know fundamentals. So chromosome. So what is chromosome? Yes, friends. Anybody in the chat box? So here, friends, chromosome. Let me paste this. So yes, it's visible. So friends, here if you see, nucleus is cell is the our body made up of cells, right? So inside the cell we have nucleus. This is the pink one, and inside that nucleus we have chromosomes. This is the chromosome. So if we observe, this is the chromosome like this one, and in the top this is one. So generally it is a short. This is a very short, like short arm. Generally people call short arm, and this is the long arm, long arm, long arm. So in the chromosome, friends, we have DNA, we have DNA plus some proteins. Some proteins. Chromosome is made up of DNA plus protein. So chromosome is the packed. It is a packed or pack of both DNA plus proteins. We all know DNA stores information as well. That's why chromosomes also stores information as well as protein. It helps to growth the cells. So these are the fundamentals. So whenever we are doing some gene therapy, right? So whenever we are doing some gene therapy, we will do either in DNA directly or on the chromosome. So that's why these two funda are very, very important. So, right. So in the gene therapy, we have different techniques. So if we see some of the concepts, what are the different techniques? Everything we will see here. So let us take this one. Yes. So friends, I hope this is visible. So these are the some of the techniques available in the gene therapy. So first one is the gene replacement. First one is the, sorry for the disturbance. So it is not copied. Let me try this one. Okay, friends, it is not working. So we will see ourselves in writing. So first one is the gene replacement. So whenever there is a defective gene replacement, whenever there is a defective gene, we are just replacing with the correct one. That is gene replacement. Gene editing is the another technique in the therapy, gene editing. So here we will edit particular malfunctioning or defective DNA. We will edit. We will do some edit with the help of CRISPR-Cas. Most important topic, CRISPR-Cas. So with the help of this CRISPR-Cas mechanism, we are going to modify gene. That is the not useful gene, or we will do some alteration for that gene. That is known as gene editing. Gene silencing, another important technique, gene silencing. It means we are making unwanted gene as a silence. So unwanted gene as a silence with the help of RNAi. This is the method or technique we are using. RNA interference. So malfunctioning gene we are going to make as a silent. Gene augmentation. So if you want new characteristics, right? So new traits for a particular gene, we are going to do some gene augmentation. We are going to enhance the gene. So these are the techniques available in the gene therapy, right? So friends, still know any questions? So let me see in the chart. Yes. Yeah, good to go. Yes. So now we will see some of the applications. So in the heading, we will see some of the advantages or applications, right? Yes. In the gene therapy, we have major applications with respect to healthcare. So which type of applications we are witnessing right now? So right. So we are going to treat genetic disorders. So genetic disorders. We are going to treat this one. So if you see, hemophilia is a rare disease. Moreover, it is a hereditary disease we have discussed, means genetic disorder. So hemophilia, whatever we are discussing, then right. So friends, you can type your answers also. Cystic fibrosis, sickle cell anemia, sickle cell anemia. These are the genetical disorders or genetical diseases. In healthcare, we have another application, that is treating the cancer cancer disease. So here we are using CAR T. So CAR cell therapy. So CAR cell therapy, it is going to modify the cancer cells and making that destruction of cancer cells. So CAR therapy is very, very important. And some of the infection or infectious diseases. So with the help of gene therapy, we are going to challenge infectious diseases. So like HIV. So it is under trials, but we are working on that. So that are the advantages with respect to healthcare. And second most advantage is industrial with respect to biotechnology. Friends, both are very correlated, healthcare and industrial application. Industrial biotech applications. So like vaccines. So with the help of gene editing and gene therapy, we have produced enough number of vaccines for COVID-19, some other vaccines, and drugs, right? And if you see biofuels, so nowadays we are moving towards green energy or renewable energy. So by modifying the microorganisms, we are trying to find biofuels which are cost effective. So modification of bioorganisms and ultimately achieving biofuels and bioplastics. So by modifying bacteria, we are trying to produce biodegradable plastics. Bio term is biodegradable plastics, right? So if we see another application in the sector agriculture, how these gene editing and gene therapy concepts are useful in the agriculture, we will see. So if we see, right, crop improvement, crop improvement, especially drought resistant, because in India we have droughts, and pest resistant, pest resistant, right? So any examples for this? So we have BT brinjal, BT cotton, right? So you can code that one. BT cotton is successful, BT brinjal, right? So these are some of the examples. And a nutritional enhancement for the crops. So nutrition enhancement. So with the help of this genetic engineering or gene editing or gene therapy, we are working to produce nutrition food like golden rice. So golden rice perhaps. So because golden rice, it is a enriched with vitamin A. That is the another thing. And so these are the major applications in the agriculture. Now we move to another area, that is livestock. So if we see another area, that is livestock. So in the livestock, generally cows and buffaloes have some of the diseases, like if you take bovine flu, right? So for chicken and all things, we have avian flu. Yes, friends, this is the correct. Yes, avian flu, bovine tuberculosis. So actually the term is bovine tuberculosis, friends. Sorry for that. So let me erase. So bovine tuberculosis, bovine TB, avian flu. These are the common diseases coming are witnessing in the livestock. So to counter that one, we are moving to gene editing technology and productivity. So moreover, if you see, productivity means productivity in terms of cows, in terms of meat. So if they are healthy, we are getting more produced meat. And another one is wool. What we get the output from the livestock, we can mention there. That is simple thing. So this is the super bright. So these are the major areas where we can apply this genetic engineering. And another thing is environmental conservation. So like wild animals and wild species. So NV conservation with the help of gene drives. gene drives, we are going to control gene drives, we are going to control invasive species. Invasive species we are going to control, and finally making the right balance between the species. So that is the beautiness of this gene editing. And overall we have completed the applications or advantages. Now we move to our fourth heading, that is challenges. So what are the major challenges with respect to, so friends, when you see, we are now discussing the current affairs, right? So here there are some are the more relevant advantages if particularly question comes hemophilia, right? So you need to mention some of the advantages with respect to hemophilia. How hemophilia helping, what are the advantages with respect of hemophilia? So that we can discuss slightly here. So if the question comes directly on the hemophilia topic. So friends, here if you see hemophilia, right? So generally, as per Roctavin, we have missed the topic. So Roctavin is the process or the name given by USFD, US Food and Drug Administration. So if we see the article once again, let me show you. So here, they are mentioning whenever we are going to change the defective gene on the X chromosome, that is gene therapy. So here copies of a corrected gene are introduced in the cell of a patient. That would result in the case of hemophilia, normal expression of necessary clotting factor. So far there is only one US approved gene therapy known as or called as Roctavin. Right friends, a Roctavin consists of a viral vector carrying necessary gene for clotting factor 8. So that is there mentioned. So this is the concept. So when it comes to our CMC Vellore people, what they did, they used some of the lentiviral vector. Lentiviral vector. So particularly they mentioned you need to remember this some of the technical terms, even though they are not relevant, but for MCQs you need to mention lentiviral vectors, right? So that they induced or integrated with the correct genes. So that lentiviral vectors help us to clotting the blood factor 8, right? That is the one of the thing you need to mention here with the help of this technique. And next one is this technique helps clotting the blood. So that is the one of the advantage with respect of this gene therapy what they did in CMC Vellore. Yes friends, and so these finally help us improved life quality. So in the replacement what we are doing, we are injecting the blood from the other people, but if we do this technique with the help of lentiviral vectors, we are directly clotting the blood, right? So this is the thing you need to mention some of the better approach, right? So moreover it is broadly applicable to all people. So sometimes if we do replacement therapy, it may not be good for all people. Sometimes it will give some autoimmune things and some other side effects, but broadly it is good. This therapy what they are saying, broadly good. So these are the advantages with respect to our current topic. So now we move to gene therapy disadvantages or challenges with respect to gene therapy, we will see. So what are the challenges friends? First, you need to mention whenever it is a biotechnology topic, so you need to talk about the ethical consideration. That is a good, right? So that you need to think, and societal. or social consideration you need to talk about, right? And third one is, it is the financial things, like financial, because the things research and everything is the cost, it is not affordable, so you need to put that one, right? So, and time taking, so these are the time taking, so that you need to mention. So time taking, these are the challenges existing in our gene therapy. So then what is our approach towards to counter this one? So we will discuss with the help of our structure, that is government role, because it is the upcoming technology and it has some challenges. What in that situation, what government should do? So government should provide their support, right? So government should provide their support for what? For research. So R&D they should give. So they should give moral support to some of the institutions like CMC Vellore, and they should give financial thing. Generally we have less spending on R&D, so that spending should be increased in terms of GDP, right? So that we should quote there, and what the government should do and what we expect from the government, like policies, better healthcare policies, because government always gives, takes or brings the policies for the people for good governance. So better policies we need, and if there like subsidies, so that you can, it is general but you should quote, because there are no much points to discuss here, so that's why we need to move away from the topic. Now, aspirant views. So being a medical student or a medical advocate, so you can talk about pro or positive for the this type of research, like gene research and everything. You should talk like positiveness, enhance the gene thing. At the same time you need to ensure the ethical implementation. Being a policy, being a reformer or a policy enthusiastic, you should talk like this. So better policies that should promote for affordable healthcare, like this you can, because affordable, because the healthcare system is the more costly right now, so you can talk as a policy into. Being a equity advocate or social equity advocate, so equity advocate, how you should talk, you should talk like uh, it should be accessible for the marginalized people. Being equity means you need to think the people like marginalized communities, that should be accessible by these people, as well as generally we can talk about rural, urban, and rich, poor. These are the equity things, right? So that is the thing. So finally you should come to the conclusion, which is the balanced conclusion. So what is the balanced conclusion we should take here? So you should code the challenges. So here, if you take the from life management, generally we are promoting replacement therapy for the life management. It is a cost. It takes around about, as per article they are mentioning, $3 lakh in 10 years for treating the hemophilia with the help of replacement therapy. Just they are mentioning the life management. So that is the very cost things. But now we are offering the, what is the solution? We are offering for the transformative solution like gene therapy, and they are mentioning that lentivirus transformative solutions they are offering. That is a good thing, and I hope it reduces the cost and time also. And but here if you take these things, not affordable, even though they are less thing, they are not affordable and it is not a scalable, as well as a we should check the safety. So you can address there are challenges like scalability, affordability, safety, long-term safety. So these are the concerns that should be addressed, and finally these things will be addressed with the help of how we will code, we will conclude like. So we need research, and government support, and collaborative with the international institutions, right? So international institution that will help us to bring more better solutions in terms of affordability and accessibility. Like this we can conclude. So friends, it is the more technology issue, so there is no need of people participation. So generally we will quote like participation of civil society, we will think, but no need to mention here, but it is not relevant. So that is the thing. So I hope you enjoyed this concept from the article, as well as the fundamentals and general things. I hope you enjoyed. So now we will move to some of the questions, relevant questions friends. These are the not previous questions. So we will see some of the relevant questions. What is gene therapy? Very basic questions if you answer the definition. So gene therapy means just modification or alteration or replacement of a malfunctioning or unwanted gene with the desired gene or corrected gene. That is the gene therapy. So in the gene therapy, we have seen four types of mechanisms like gene editing, gene augmentation, right? So all we have seen there. So you can also write just definition and some types. Leave that part. Now another part is discuss its potential in treating hereditary diseases like hemophilia and ethical and societal implications. So friends, you can quote the relevant points from the article, whatever it is more relevant to our article. So you can think about it one and comment your answer below the video. Next question is, the use of gene therapy raises concerns about the misuse of designer baby. So friends, it is slightly out of the topic, but so being a this type of research, so it we need to address ethics. So the question comes in like this one. So what is designer baby? So designer baby means we are designing the baby before birth. Designing. So what are the traits? How face look, how lips look, how eyes look. So these type of genes we are determining and we are inducing into the baby. So it is a more controversial topic. So that's why they are mentioning misuse. So just think about it and discuss ethical considerations and regulatory measures need to address this one. So just think yourself. So here we will see some of the MCQs. Gene therapy has potential in treating which of the following disease? Sickle cell anemia, hemophilia, cystic fibrosis, and Alzheimer's disease. So, friends, what is your opinion? So, generally people go to 1, 2, 3, 4. Friends, not 1, 2, 3, 4. Here, 4 is not a correct answer. It is the in the Alzheimer's disease. It is the not completely evolved. It is in the trials. So, answer would be 1, 2, 3. Consider the following statements about chromosomes. Friends, it is the fundamental questions. So whatever the gene therapies we are doing directly on DNA or either chromosome, because chromosome is the pack of DNA plus proteins. So human has 23 pairs of chromosome including 22 autosomes and one pair of sex chromosomes. So friends, I will discuss in detail about more here. So generally this is the correct statements. We have 46 chromosomes, that is 23 pairs. So here dogs have 39 pairs. So dogs have 39 pairs. Rice plant have 12 pairs, right? 12 pairs of chromosome. So what is chromosome? We all know chromosome is the pack of DNA plus protein. So in human we have 23 pairs like XX, XY. So XX denotes female, XY denotes male, right? So here if Y is the determining factor for either male or either female, right? That is the most concept, right? So chromosomes are made of DNA and protein. That is also correct. During mitosis, chromosomes are replicated and distributed to daughter cells. So friends, all are correct. So here we have come across new term, that is mitosis. There are two terms like mitosis is the one thing and meiosis is the another thing. So meiosis is helping in the reproduction. Right here is the replication and distribution to daughter cell. This is the difference between meiosis and mitosis. These are the cell division mechanisms in the chromosome. So friends, final question for this topic is primary role of Cas9 protein in CRISPR-Cas system. Friends, I advise you or suggest you to deep dive this CRISPR-Cas mechanism in detail. It is a burning topic, right? So just read the answers. To identify the target DNA sequence, to guide RNA molecule to target DNA, to act as a molecular scissor to cut DNA, to repair DNA after it has been cut. So friends, Cas9 protein act as a scissor, right? It acts as a scissor. So friends, try to see if you observe the diagram also in my thumbnail. So we have mentioned some type of scissors. So that scissors are related to Cas9 protein. So scissor to cut the DNA. It cuts the DNA and replaces with the help of other RNA guided mechanisms. So friends, I hope if you have any questions, let me know in the chat. Thank you so much. Friends, thank you so much for watching the video. And if you have any doubts, you can ask me by logging into the link below the video. I'm giving some of the links. There you can check the links and you can straight away contact with me. And you can also get these PDF links, handwritten PDF links, everything I am posting there. So thank you so much for watching the video. We will meet with the another video.